Thank you. I want to thank you first of all, for the wonderful introduction and for the invitation. And I want to thank you all for staying on the last day of conference to hear it. I have some other organizations that don't quite like me as much, and they always put me last, assuming that everybody would be gone. So I'm challenged today because here's the title, Connecting STEM Research, Teaching, Learning, and National Advocacy to Real-World Challenges. And no offense, but I didn't write the title. If I did, I would have called it something like, how in the world are we supposed to do all that? Let alone, how am I supposed to talk about all that in 45 minutes? But I'll do what I can. Before I do, though, I want to introduce my grandmother, who passed away about 10 years ago now. And I'm thankful, first of all, that she passed on to me her love of education and her desire to make the world a better place before she passed on. But I'm also thankful because she taught me something that you can't learn at places like Harvard and Berkeley, and that is, if you want to get a point across, just tell a good story. So I don't give talks, I don't lecture, I don't teach, I just tell a story. I guess the more pedagogical way to say that is learning and teaching by experience. I want to start, before I tell my stories, I always do my acknowledgements first. That way, if you do decide you want to leave, you've seen the important stuff. This is a proverb that I learned while working in Zimbabwe, and I won't try to pronounce it, but loosely translated it means people are people through other people. And so I always start by thanking my family, because I am who I am because of these guys. This is an old picture. They won't let me be in them anymore for obvious reasons. But I, especially my mom and my dad, Romeo and Susie Hayes, without them, I'm a biologist, so without them, I literally wouldn't be here. But also my wife, Catherine Kim, for her love and support, and without her, these two wonderful people, Cassina Simone Kim Hayes and Tyler Casey Kim Hayes, wouldn't be here. And this is an old picture too, but I show this one because these are their prom pictures. And I think it epitomizes the love and relationship that we have. For example, I don't know which made me more proud, that my son borrowed my tie for his prom, or that my daughter borrowed my earrings for her prom. After 18 years of, no, daddy's earrings are too grown up for you, sweetheart, she finally got to go. And as I said, I always do my acknowledgements this way with family first, but also this is kind of appropriate for what I've been asked to speak on here today, because what you've really asked me to talk about is conflict and conflict resolution. How can we do research? And if you're like me and you do research in the field and the lab, there's a conflict there because you can't do both. You got to do one or the other. There's a conflict on the job because you got to do research, you got to do teaching, and if you care about inclusion and diversity and public service and reaching out, how can you possibly do all those things? There's a conflict, and on top of it, how can you do all those things on your job and then still be true to your family, especially if you have kids or others that you have to care for? The answer is you get rid of the conflict. And some of the ways that I got rid of conflict, I don't know if we can have the lights down, this looks a little dark. Some of the ways that I got rid of the conflict was, here I am doing field research, and I had options, and those options were to leave, in this case, my 2-year-old son at home, which puts more of a burden on my wife, or the options were to have a great father-son time out in the field. And several things are happening in this photograph. One, there's my son having a great time. Two, there's my graduate student and a postdoc also seeing that just because you're committed to your job doesn't mean that you have to sacrifice family. So there's that old saying of don't bring the office home and don't take home to the office. I don't think so. Here's another example, and several things are going on here. Here's my 2-year-old daughter. I don't know if we can have the lights down a little bit so you can see this beautiful young woman. And several things are going on here. One is, for the longest time, many of my female students wouldn't go on field trips because they didn't want to sleep outside and things like that. And now they've got to grow up with this strong young woman here. And again, daddy-daughter time. The other thing I think that happened here that eliminates some of the conflict is, there I am doing research, and I'm with my family, and I got a great lecture out of it. Because see, my lecture on osmoregulation now is entitled, Why My Daughter Doesn't Pee in the Desert. And so instead of just having to hear about kidneys and arginine vasopressin, they get to hear this story about my daughter, and I never had to change her diaper, and because she didn't pee, because once she perspired and she got dehydrated, then the antidiuretic hormone kicks in. It's a much better story, and also allows my students to see me as a person with his daughter, and they get to learn probably the most boring topic that I have to teach in that course. And also, the same way that my family made me, I like to think that I made them. You saw, let me go back. You see her climbing, she's about 15 feet up there. It probably contributed to her being a nationally ranked gymnast. So there she is doing things that I could never watch but could photograph with my eyes closed. And she's now in college studying neuromuscular interaction, probably related to athletics, and having grown up with Dad in the lab and in the field. So those are just some of the ways that I think I've sort of eliminated that conflict and made it work out for everything. Continuing with my acknowledgements, here's my funding. I learned a lot from some of these guys too. These guys in red are the chemical company that no longer want to talk to me. But they're the ones who really taught me about advocacy, as I'll tell you. And here's all the students that have worked in my lab over the last 20 years and published with me. Everybody in blue is an undergraduate. I had a wonderful experience as an undergraduate myself, and for everybody who says you can't do that kind of work with undergraduates, I would argue that my fourth-year undergraduate, who's been in my lab since they were a freshman, is as good as anybody's first-year graduate student. Here's my current list of students, and here's a photograph that sort of addresses also some of the things I'm supposed to talk about. There is one guy now, but at this photograph, they were all women. Everybody, 7 of the people in this photograph speak English as a second language. All but one is a first-generation, low-income student, and 3 of the people here grew up in families that worked in agriculture and have actually been sprayed and exposed to many of the chemicals that I work with in my laboratory. So that's been some of the draw that bring them into the laboratory. I've also seen a shift from students attracted to my laboratory because they, like me, are just really in love with frogs and animals, to students who are now learning about animals and animal biology because they're interested in public health and human health. And I'll kind of show you how that transition happened. So the story I'm going to tell you, I call From Silent Spring to Silent Night, a tale of toads and men. And it's not just a story about research, it's about research that incorporates teaching, that incorporates undergraduates, that incorporates diversity, and at this stage in my career, really incorporates advocacy and public service. The story, and I'm going to tell it to you in chronological order, exactly as it happened, starts with a little boy who likes frogs, period. And it will end exactly the same way. I have to go back to my grandmother again, because much of my story starts with my grandmother. And here's her house. It was a plywood tin roof house. She was still living in that house when she passed away. Her grandfather built that house. Her grandmother's bill of sale from where her grandfather bought her freedom was still in that house when she passed away. So a huge amount of that history, a huge amount of who I am was in that house. And importantly, every weekend we went to that house to stay with my grandmother, and there was a huge forest behind her house that I spent hours and hours getting lost in. And now Google Earth, I can bring that to you. And there's the house, there it was. The cemetery was always there, the highways were there, the horse pasture was always there. It was just that tiny little strip of woods, but I remember it from my childhood as being like the Amazon. I remember being lost in there for hours with birds and snakes, and of course, these guys. And one of the things that fascinated me about frogs is the altruism. Many of you might not know about this, right? You see this frog on the top? He's hurt his leg, and the one on the bottom is nice enough to give it a ride home. Those are just some of the things that I observed as a child that fascinated me. And you laugh, but I have students who've actually for several months thought that was true and then said, oh, that's not what they're doing. What they're actually doing here is on the way home from this ride, the females drop off some eggs. And each one of these little clumps, these are wood frogs, each one of these little clumps is about 2,000 eggs by a single female, but hundreds of females might lay their eggs in these huge communal clumps that you see here. And what fascinated me about this situation, remember, I'm a little boy who likes frogs. I'll tell you how old in a minute. Is that the eggs in the center could be 10 degrees warmer than the eggs on the edge. That means that these females, by getting there first and laying their eggs in the center, could manipulate things like how fast the larvae develop, how fast they metamorphose, how fast they grow, because temperature will make them grow and develop faster. But also in some reptiles and fish anyway, temperature can also influence sex. So I was interested in whether or not the animals in the middle that were warmer might be more likely to come out. I'm sorry, you're going to see a lot of gonads in this talk. These are testes and these are ovaries, and I did these experiments some of which started back in South Carolina. But I did these experiments where I asked, does temperature affect the sex ratio? And I was also interested in the interaction between genes and the environment. So I looked for sex chromosomes and the idea that you might have sex chromosomes that might limit how much the environment can influence whether or not you become male or female. Now, here's where we're at in my story. This is where I usually tell undergraduates, I like to give them a little bit of a history lesson. This is where I explain to them that this is work for my undergraduate thesis. This is back where cut and paste, literally meant cut and paste. I have to tell them that function that you have in Adobe Photoshop, cut and paste, that's where it came from, because we had to cut the stuff out of the picture and paste it on the page. And in fact, this is work from my undergraduate thesis, the summa cum laude that you heard mentioned. And here I am. This happened at Harvard, right across the bridge. I was age 19 there. There's my professor, Bruce Waldman. And as a first-generation, low-income minority student, this was an incredible experience for me as a little boy who likes frogs. One is, I always tell people, take your disadvantages and make them into advantages. I was there because I was on work study and I had to work to pay for my books and things. And I got this wonderful experience starting my freshman year from a guy who, for whatever reason, treated me like a graduate student. And I always say, if it wasn't for this man and for my wife, I never would have finished. It was such a foreign place to me, being up north, being one of, at the time, it was 16% minority. And I always tell people from California, that included Asians, that there was just so little there that I could identify with, and this happened. This also the place where I learned what professors really did, because all I know was the teaching, and now I got to see and have this experience where I could see what actually happens when he's not standing behind the lecture, and that's what I fell in love with, was the research aspect. I never thought I would enjoy teaching until I actually had to do it. I guess this slide also illustrates that sometimes it's good to find out what you don't want to do as well. Because you can probably tell by the look on Laura's face that she went into something else, because she was not as happy as I was to be in a swamp at 6 a.m. on a Sunday morning. But for me, it was a dream come true. That dream led to another dream come true. And it's one of the things that I stress with my students. Think about the thing that if you had nothing else to do, what would you do? Make that your job. And one of the dreams that I had, and the reason I started working there to make extra money, is that I dreamed of going to Africa. I remember as a child, my dad bringing home these National Geographic magazines, and I remember folding out the pages and looking at the maps and dreaming of going to this magical place. And it was a dream. My father made $9,000 a year for a family of 5. First time I ever got on a plane was when I came here to go to Harvard. It was completely out of the realm of possibility until graduate school. So here's where we are in my story now. Little boy who likes frogs in my grandma's house. Now I'm in graduate school at Berkeley. I got to grow this weird beard, but I not only got to go to Africa, but National Geographic paid for it. I got to be in the magazine, I got to be on the show. This was truly, truly, if you could imagine what it means to have a dream come true, I became that guy that I watched on television as a little kid. And again, relative to the questions that I'm supposed to address here, this was one of those things that's not so easy to do if you never see anybody in a magazine or on TV or talking about science or doing what you want to do who looks like you. I didn't have that. I got to work in the Arabuko-Sokoke on the coast in Kenya, and one of the cool things about working in Africa is you get to say things like Arabuko-Sokoke. It's not something that everybody gets to say. But in the Arabuko-Sokoke, I discovered actually, and started working on this frog. And what was exciting to me, little boy who likes frogs, is that the males and females look like they don't belong to each other. Very unique. And I got interested in ecologically why they're differently colored. What are they doing differently in their environments? But I also got interested in how they get to be differently colored. And what I discovered was they all start out green. So this is after Adobe Photoshop, so this cut and paste looks a lot nicer. Here's a control photograph once a day for 6 days. It's the same animal. They all start out green, but the females change color at sexual maturity. So I worked with an undergraduate and a graduate student. And one of the things for me, I like to see things. And this is one of those things that is easy for somebody to see and develop hypotheses, right? And in this case, our hypothesis was that estrogen caused them to change color. In the same way that estrogen causes girls to grow breasts when they reach puberty, we hypothesized that our frogs change color because of estrogen when they reach puberty. It was a really simple hypothesis. And the test was simple. We just dipped the frogs in hormone solution, literally, and we found out that, sure enough, the estrogen-treated ones changed color, and then we published a nice paper on it. Karen Menendez and Nigel Noriega and I. And then something weird happened. It happened on February 15th, 1993. And I remember the date exactly because it's the day before my son was born. He grew up to look a lot better. I thought he was beautiful, though, so I'm convinced now that there are pregnancy hormones in men, too. Because he had zits and a cone head and all, but he was beautiful to me. It was the most beautiful thing I'd ever seen. And that's why I remember the date. But what happened was I was at UC Davis and giving a talk, and my wife, who was about 9.5 months pregnant, was in the audience, and she started going into labor. See, there's that thing again where you mix work and home. She started going into labor in my talk, and as we were driving down the road to get back to Oakland where my son was born, my wife says, between contractions, "You should patent that frog." And I thought it was like the crazy hormones. And I said, "You can't patent a frog," but we did. Here's the frog, Hyperolis argus. We call it the Hyperolis argus endocrine screen, or the HACE test. And I patented this actually with the students who helped discover it, Nigel Noriega and Karen Menendez. And let me tell you first, before I tell you how things got weird, here's why you patent a frog, first off. The controls are always green. And then working with Nigel Noriega, my graduate student at the time, we screened a whole bunch of compounds, right? Estradiol, that's the natural estrogen. Doesn't matter if you're a frog, a dog, a cat, a hog, or a human. If you're an adult female, this hormone circulates in your body. Ethinyl estradiol is the synthetic estrogen used in birth control pill. It made my frog change color. DES is a pharmaceutical estrogen. It made my frog change color. And I could call it my frog because I patented it. DDT, a pesticide, made my frog change color. So we patented the frog because it was a screen. Because you see, every estrogen that made my frog change color was also known to promote breast cancer in humans. In addition, we showed that we could treat these frogs with tamoxifen and block the color change, and tamoxifen was the estrogen blocker used to treat breast cancer in humans, so we could also screen new pharmaceuticals that might then go on to be used in the treatment of breast cancer. And so I patented this model for that reason, and it also then started to attract a whole different set of students who are now interested in human health, as opposed to the students who are interested in collecting the frogs and being in Africa, but that's okay. We still have good conversations. Here's where things get weird, though. Here's where a little boy who likes frogs gets introduced to grown-up words. And for some reason, I think grown-up words always come in two. In this case, the grown-up word was intellectual property. See, the university said, yeah, that's a great idea, but if you don't make money on it, we're going to sell it. It's ours. And so here's where things get weird, right? I got asked to study this chemical atrazine, which you heard about in the introduction. Here it is. I hated chemistry in college. In fact, it was so nice I had to take it twice. But I can always tell people I did not get an F. That's because Harvard only gives A, B, C, D, E, and I convinced my parents that the E was for excellent. They wanted me to come back. Now, you wouldn't believe it. I have to do so much chemistry. It's incredible. You can survive, I guess. I tell students, don't worry about it. You did bad in one class. So anyway, it's an herbicide that's used on corn. It kills weeds. It's been used since 1958. We use 80 million pounds annually in the United States. It's used in more than 80 countries, but now it's outlawed in all of Europe. Now, full disclosure, that's not true. The company lawyer writes me all the time. They said it has not been banned in all of Europe. It has been denied regulatory approval by the European Union. I'm not sure what the difference is. All I know is that this one pisses them off, so that's the slide that I use. You know, that's just the kind of brother I am. So they asked me to examine the African clawed frog. They said, because the African clawed frog, the Xenopus, is very commonly used in biology. And the reason why, I like to give people history. You know why this frog is used? For a weird reason. In 1920, somebody discovered that the human pregnancy hormone, HCG, will make this frog lay eggs. So by 1940, history is important. By 1940, this frog became the pregnancy test. If you thought you were pregnant in 1940, you go to the doctor, they inject some urine into the frog, and if it laid eggs, you're either happy or sad, depending on your situation. So I tell this story for three reasons. I always tell my students, if you show a slide, you have to have three good reasons for it. One is, it shows you the value of curiosity-based research, right? Like, who's the first guy who thought, hey, I wonder what will happen if I inject pee into a frog? Like, how do you come up with that? I've searched and searched. I can't figure out how anybody decided to do it. The second reason I tell you is it helps people understand that just because you're not doing directly human-based research doesn't mean that it doesn't have human implications. In the same way that the estrogens that promote breast cancer also make my frog change color and are useful that way, I'm interested in the frogs, but the human pregnancy hormone is so similar to this frog's hormones that it'll make it lay eggs. That's why I call the talk A Tale of Toads and Men, because many of the things that we discover in frogs are transferable to humans, and it attracts a different set of students when you can make them see that. Oh, I said 3 reasons, I guess. The third reason is, you saw that picture of me with my son on my back, that was in San Diego. Turns out when they developed new pregnancy tests, people just threw these frogs out. So instead of buying them from a store or from my guy in Woodland, I can just drive to San Diego and collect them on my own. It makes for a good outing, which I guess technically makes my frogs African-American clawed frogs, but that's sort of an irrelevant point to the story. So we discovered, while I was being paid by this company, that atrazine inhibited growth of the voice box of the larynx in males. And that's bad news, because male frogs sing and females don't for the same reason that men have deeper voices than women: testosterone. No big multi-billion-dollar company wants to hear that their number one selling chemical messes with testosterone. And I told you there'd be a lot of gonads here, but what we discovered is we went to the gonads to see what was going on, and here's an animal that has testes, and then ovaries, and then another testes, and this frog could hurt its leg and give itself a ride home, which is not normal. And I say that, and that's not a judgment. I say that because there are fish that are naturally hermaphroditic. There are no frogs that are naturally hermaphroditic, despite what you may have heard or seen in Jurassic Park. Frogs are not naturally hermaphroditic. I found out that that's why people think that. So here we decided to go to the testis and ask this question. The testis should make testosterone. You know what testosterone means? Most people don't. It's a portmanteau. Do you know that word? I learned that word recently. I wish I'd known it for the SAT. But portmanteau is when you stick 2 words together, like smoke and fog, you get smog. Twist and jerk, you get twerk. Testosterone, that's usually a lot funnier with my younger audiences. But testosterone literally means testicular hormone. It's 2 words stuck together, so it's the male hormone. And we propose that atrazine turned on aromatase. It's the enzyme or the machinery, if you will, that converts testosterone into estrogen, which means generator of estrus. It's the female hormone. And so your testosterone goes away, you're demasculinized, and you're subsequently feminized. And so we showed that in part by looking at adult male frogs, and here's atrazine-treated males that aren't different from females. Okay, now here's where we're at in my story. You guys know PNAS, Proceedings of the National Academy of Sciences? So we published this paper in PNAS, and I called my mom. It's a 100% true story. I called my mom. I'm coming up for tenure. That's where we are in the story. I called my mom and said, Mom, I have a paper coming out in PNAS. Silence. I said, Mom, she said, yeah, I heard you. I just don't understand how you get a paper cut on your penis. And I said, no, PNAS. And my mom, this is a 100% true story. My mom, and this is going to get at the advocacy question, you'll see. My mom, she calls me back the next Sunday, and she said, how important is that paper? I said, really important. She goes, well, I went to Barnes & Noble, and they never heard of that magazine. And I'm going to tell you why that's important. So now this is my most important publication. It's a kid's book. Somebody just actually asked me to sign. I didn't even write it, but my mom can get it in Barnes & Noble, and it allows me an avenue, a way to reach out and talk to young elementary, middle school students. And now they're hearing about science through this wonderfully written book. And my mom can buy it in Barnes & Noble, and I'll get back to why that's important. Hey, by the way, this PNAS paper, 4 black men and a Latina co-authored this paper. It's probably a record for the National Academy. I'm proud of that. As proud as I was, and I did get tenure, by the way. Some would probably, well, let's not go there. As important as it was for all those reasons, it didn't answer a couple questions. Like, one, we didn't know if these hermaphrodites were males with ovaries or females with testes. Sounds like an easy question to answer, but I discovered with all my cut and pasting at Harvard that frogs don't have sex chromosomes. They might have sex-determining genes, but they don't have chromosomes. And we also didn't know what happens when these animals become adults, because we only looked at them right after metamorphosis. So I designed some experiments to try to answer these questions. And one, it again, might sound like an easy question to answer, what happens when they become adults, except that what I'm not telling you is it takes these animals 4 to 5 years to reach sexual maturity. So you got to get an undergraduate or a graduate student in their first year and say, I got a project for you. And maybe by the time you graduate, in fact, it was worse than that. It took 8 years, but people still graduated. I'll tell you about that. And here was the answer. These are adults. This guy who looks like he's smiling there, they hurt their legs all the time. By the time the paper came out, they had discovered a gene that only females have. So this is PCR analysis. And we could tell then that he's a male, and that's his brother. And not only did he behave like a female, he laid eggs and completely functioned like a female. And I was excited. I could have published that, right? But I had tenure. I was in no hurry. I wanted to know what happens to the 90% of males that don't completely turn into females. Are they normal? Can they function like a female? And so I wanted to do these studies where I looked at whether or not they could reproduce, whether or not they could breed. The problem is these frogs like to breed in the spring. And as you may know, unfortunately, so do undergraduates. They all want to go away for spring break, some kind of pool party thing. So the trick is now we're talking about inclusion. How do you get a bunch of undergraduates to stay around for spring break and hang out in my lab? So I devised, now everybody might not be able to use this. Spring break 2008, I said, I will throw you a pool party. Snoop Dogg won't be there, but I will throw you a pool party if you stay behind, and you'll get a PNAS paper out of it. You got students who do this, right? Okay, 100% true story. It went something like this. This is what we call the apparatus. I'm not joking. This is published in PNAS. We put 4 female frogs in there, 4 control males, 4 atrazine-treated males. I know this ain't the sex ratio the young people want at the club, but the idea is the females eliminate. We want to see if these guys could compete. And that's just what we did. I just made it up off the top of my head. And so we did it, and here's what this looks like. You put them in there. This is exactly how we did this. See, there they are. They're swimming around. The lights go out, you put on a little Marvin Gaye. And then you come back the next morning and you just look at, as the young people say, you just look at who hooked up and who didn't. And we have stitches in there so we could tell which frog was which. And so we did this really simple experiment, and it turns out the atrazine-treated males almost never win. Only 2 cases out of 4 trials did they win. And then we looked at the hormones. So I'm an endocrinologist, so I can't just look. Imagine you're in the club, somebody comes and starts taking down names at the end of the night to see who's with who. And then they pull you apart, stick a needle in your heart, and take a blood sample. That's how this went down. And so, because they don't have veins, you got to get them in the heart. It doesn't hurt them, though. They like it. And so it turns out, as you might guess, the controls, the unexposed animals, have, on average, higher testosterone. But what's important is the individuals who end up with the females, there's the 2 with the atrazine, have much higher testosterone levels. So either these guys don't have enough testosterone to attract the females, or they get beat up by all the other males with testosterone. Well, then we did something called the Motel 6 experiments. And in this case, there was no competition. We just got them a room. It looks like this. We put them together. And we asked, how many eggs can you fertilize? And again, there's no competition in this particular test. And when you do that, here's what it looks like. So these are about 2,000 eggs from a single female. And so you collect those the night after. And then you just look at how many hatch. So these are various developmental stages versus how many are not hatched or are not fertilized. It's a very complicated procedure. Yeah, there's an undergraduate sitting in the lab every day going 1, 2, 3. He went into math after that. It turns out that if you do that, the control males fertilize about 85% of a female's eggs. The atrazine-treated males about 15%. And it's not just because of the behavior, but that's part of the problem. One is they just sit there and watch the females. They don't even try, which is probably why they lose in the pool party. But the other reason is, and I told you I like visuals. The other reason is that this is what the testes of a control looks like under the microscope, and this is an atrazine-treated one. And what was beautiful about this study was the person who did all the raising of the animals and things was different from the students who did the behavioral tests, which were different from the students who did the histological analysis. So we have a group of students that, if you're interested in development, or if you're interested in behavior, or if you're interested in histology, looking at tissues under the microscope, if you're interested in molecular biology, we had all these things going on in the lab. And what was beautiful about this is I could take these photographs and give them to brand new students and say, separate them in the 2 piles. I mean, we did all the statistics and stuff, but I like to see differences. And what you're looking at is controls with sperm filling up the testicular tubules and atrazine-treated males with just cellular debris. A very easy thing that you can train students how to do. So we got a second paper in PNAS. We called it Atrazine Induces Complete Feminization and Chemical Castration in Male African Clawed Frogs. The company hates the term chemical castration. You know it's a grown-up word because it comes in two. And it sounds even worse when you say it like this, chemical castration. The company hates it. That's why I put it in the title. That's just the kind of brother I am. You know, the other thing that's important about this paper is there's 1, 2, 3, 4, 5, 6, 7, 8 undergraduates co-authored this paper. Every one of them now has either an MD or a PhD, and the graduate student who was on it was a Morehouse man. Again, something else. A Morehouse man that I recruited to my lab when he was an undergraduate. Very important to me. So then we wanted to know, well, is this something that happens to all frogs, or is this just some weird thing that we only see in these African clod frogs? And so we did these studies, and this paper was published in Nature, by the way, and by now I'm coming up for full professor, and so that was significant in that decision. So these are testes, and then these are all eggs that are bursting through the surface of this male's testis. I call it the junk in the trunk. That shouldn't happen. You shouldn't have eggs in your testes. And now at this stage, now we're going to try to get into the advocacy thing a little bit, because at this stage, I'm now interacting with the Environmental Protection Agency. And I always say curfew, I probably did that too, but I mean embargo. I broke embargo because I sent this to the Environmental Protection Agency. I said, look what atrazine does to these frogs. And they sent me back an email. They said, well, that's interesting, but that's not an adverse effect that would trigger us to reconsider or reassess the chemical in the environment. It's not an adverse effect, they said. Now, my wife tells me that there is nothing more painful in life than childbirth. She's going to kill me for showing this photograph. I got to give her that because I'll never experience that. But I would guess that a dozen chicken eggs in my testicle would have to be in the top 5. But the EPA says, no, it's not an adverse effect. Yes, you can have eggs in your testes. It's okay. I guess, I don't know. So the reason that we did these North American leopard frogs is we wanted to see if these effects occur in the wild. And so here's a testis from a wild collected animal. Now we're out of the lab, so now imagine I take a van with a bunch of students. I'll show you where we went. And we found things like this. I'm going to slice this up. So this is what my students get to see under the microscope. I'm going to fold out a slice. I'm going to blow that up. I'm going to blow this up. These are testes, testicular tubules, just like we have, except these are eggs in the testis instead of sperm. When we published the paper, I call them testicular oocytes. Testicular oocytes. You know it's a grown-up word because it comes in two. Testicular oocytes. The lawyer wrote a letter to Nature and to my university. They wanted me to have me fired and retract the paper because they said I made up a word. Aren't all words made up before they're words? Plus, I told them I went to Harvard. I can make up a word. Fool. So here's what we did. We found out these animals had these testicular oocytes, and we looked at a map of the United States. Here's where most of the atrazine is used, down to gray-white. And we did this, I always tell people, where we control for latitude, but that's a lie. This is Highway I-80. And we were going to a conference in Indiana, and so we collected water and frogs along the way. Me and a van full of 9 students, and we got a Nature paper out of it. See, that's fuel efficiency. And I got promoted. But the reason we got promoted is not just because of the Nature paper. It's in part because we went on to show how important atrazine was in the environment. And one of the other things that I think is important with students is sort of to appeal to pop culture, if you will. And so the next trip we went on, I said, CSI, we're going to go find out who the real culprit is in Nebraska and how important atrazine is in Nebraska. And the reason that we had to look at how important atrazine is when you find, in the lab it's one thing because it's a controlled study, right? But in the field, there's always other suspects. So this is just all the chemicals that are used on this one field alone. The herbicides are in red, the insecticides are in purple, and the fungicides are in blue. And now you see why there's so many undergraduates. We did this huge study where we studied each one of those chemicals alone or in combination. And the colors you see are because I do my experiments. I used to say double blind, but then somebody pointed out that double blind means that the person receiving the treatment doesn't know what it is. I guess it's still double blind because my frogs don't know what they're being exposed to. But the point is the students only know to add the red, yellow, red solution to the red, yellow, red tank. Who's treated with what is not revealed until after we do the analysis. So it's all done without bias. And the short version of the story, and this is the next to the last frog story I'll tell you, is that we found that when you mix these pesticides, they increase stress hormones, and among other things, cause really retarded development and growth, which is bad if you're a frog. We then went on, and this last frog thing isn't published, but we then went on to study frogs in Salinas, and the Salinas River flows south to north, And the arrow is red at the end because there's a lot of agriculture once you get to the north. For example, who's ever eaten anything from Salinas? Everybody raise your hand, because 85% of this country's greens are grown in Salinas. 85%. I guarantee you've eaten something from Salinas probably every day of your life, unless you're like one of my students and you only eat meat. And then even that probably came from Salinas. So it turns out that there's a natural experiment, because when you're at the flow of the river, the water's nice and clean, 1.5 foot deep, 22 degrees, no agriculture yet. This is where rich people in Monterey get their water from. As you go further downstream, there's no agriculture, but all the water is being drained off for agriculture. So you have animals under stress like this, where there's like 3,000 tadpoles in about an inch of water that it's at about 100 degrees. So it's pretty stressful. And I should point out, 3 of my students grew up in Salinas. And so they're working in an area where they grew up in. And then when you get to Salinas, there's 1.5 foot deep water. There's no physical stress, but 100% of that water is agricultural runoff. I told you I like visuals, so the short version of the story, and this is stuff we still haven't published, is this is a frog, the tadpole, collected upstream of agriculture. And this tadpole, same species, same age, same stage, collected on the same day, just 2 hours drive away. That's an animal that's living in water that runs off of your food. Only difference, no difference in temperature, water height. The only difference is this animal lives in water. I can show you exactly where he came from. These animals that are small like that are surrounded by agriculture and agricultural runoff with warning, poison signs, peligro, do not enter, all over the field, yet somehow an hour later at the grocery store it's supposed to be safe. Or a plane ride away it's supposed to be safe when it comes to your dinner table. Something interesting is John Steinbeck in East of Eden in 1952 had something to say about Salinas. And I'll read to you now. And this is where I get that opportunity to tell students that even if you're interested in science, here's why you have to take all those non-science classes so you can be well-rounded. Here's what John Steinbeck said about Salinas. He said, Salinas was surrounded and penetrated with swamps, with tool-filled ponds, and every pond spawned thousands of frogs. With the evening, the air was so full of their song that it was a kind of roaring silence. It was a veil, a background. And his sudden disappearance, as at their clap of thunder, was a shocking thing. It is possible if in the night the frog song should have stopped, everyone in Salinas would have awakened, feeling that there was a great noise. In their millions, the frog songs seem to have a beat and a cadence. And perhaps it is the ear's function to do this, just as it is the eye's business to make stars twinkle. Here's what Salinas sounds like now. The students that I have that grew up there have never heard a native frog call in Salinas, and I haven't heard one in the 7 years that I've been working there. And it's an excellent opportunity to point out that even if you're a scientist, you learn things everywhere. I think that piece is better than a scientific literature that said, here's how many frogs were here in 1952. The frogs was so important that a literary artist committed a half page to talking about them in that landscape, and now it's gone. Thus, my title, Silent Night. Here's a slide showing, and I'm not going to tell you it's atrazine or even pesticides that have caused all the frogs to go away, but here's a paper I published with my graduate students showing all the factors that contribute to amphibian declines. For those of you who don't know, 70% of all amphibians are in decline. And it's not just pesticides, habitat loss is the biggest factor. But if you drain off all the water for agriculture, the only place you have as a frog to lay your eggs is in water tainted with chemicals that lower your immune function, that make you more likely to get diseases. The chemicals are playing a critical role in that decline, globally, I believe. This is a slide from Lake Nabugabo in Uganda, showing runoff from this crop that goes into these containers that is the drinking water and cooking water for this village. If I told the men in this village, the frogs and fish in that water can't reproduce and they have low immune function, they'd immediately see not the connection, but the oneness between environmental health and public health. But most of us live in situations like this. Here's Oakland. That's where the A's or somebody plays. I don't go to sports. But I live somewhere here. My water just comes from there. And this atrazine and other chemicals can be in the rainwater at levels that we know to be biologically active. So I call this from silent spring to silent night because in much the same way that Rachel Carson taught in Silent Spring, that the death of birds and the role of pesticides were a warning to us, in much the same way, I believe that the decline in amphibians, a group of animals that have been around since the days of the dinosaurs, that are now 70% in decline because of our activity, I think it's a warning to us. A colleague of mine wrote, in ecoepidemiology, the occurrence of an association in more than one species and species population is very strong evidence for causation. Well, it turns out that these effects have been shown not just in my work, but in work in every vertebrate class that's been examined. And I can show you examples. I published a paper along with a bunch of other scientists, 22 scientists from 12 different countries. We called it Demasculinization and Feminization of Male Gonads by Atrazine, Consistent Effects Across Vertebrate Classes. And this was significant because it showed how widespread the problem was. I also invented the term gonadotoxin in this paper. Lawyer wrote all kinds of notes. I made up a word again. You know, I read in Science magazine that all language, like all humans, originated in Africa. So I'm just doing what my people's good at. I don't know what his problem is. So here's my work in frogs, sperm in the testis, give it atrazine, no sperm. Here's a guy in Belgium that showed the same thing in fish. Here's a couple in Argentina who showed the same thing in like a big alligator. Here's people in Nigeria and in Croatia that have shown a rat, sperm in the testis, give it atrazine, no sperm. And here's work in birds that was done in Pakistan. So we've shown that this is a thing that occurs across vertebrate animals. And what's more is, we've shown in humans, and this isn't my work, this is somebody else's work, that in humans, men who have atrazine in their urine are less likely to be able to get their wives pregnant. They have a low sperm count. And that's just a correlation, but imagine if you give atrazine to a fish, to an amphibian, to a reptile, to a bird, to a mammal, they all suffer from a loss of sperm. And if you have this compound in your body as a human, you have a low sperm count. Now what's more is, I squashed the data down now because these are levels in field workers in California, and these are levels of atrazine in men who apply it, 2,400 parts per billion. These guys have 24,000 times the atrazine in their urine than we know is associated with low sperm count and low fertility in men. These guys have 24,000 times the atrazine in their urine than we use to chemically castrate frogs. One of these guys could pee in a bucket, and I could dilute their urine 24,000 times and use it to chemically castrate 24,000 buckets of 30 tadpoles each. Now a little boy who likes frogs gets introduced to another grown-up word, environmental justice and environmental racism, because 90% of the people, like my 3 students that I have in my lab, who work in agriculture are Hispanic or Latino. That's significant because if you look at California, a few facts you might not know, we're the 5th largest economy in the world because of agriculture. If we were our own country, we'd be the 5th richest country in the world. 1 in 10 jobs are in ag, 30% of the land is in ag. We make 350 agricultural products. Half of the U.S.'s food comes from California. Half. We use more pesticides than any other state, and 90% of the workers are Hispanic. Now, if I put in red now the top 10 counties for agriculture, these are the 10 counties to 30% that makes us the 5th largest economy in the world. Where do you think the 30 poorest towns are in California? So the people, like my 3 students who come from these areas, the people who make us the 5th largest economy in the world are targets of chemicals that we know are associated with adverse health outcomes. The other half of the equation is, does atrazine induce aromatase like it does in frogs and fish and reptiles? That's not going to make you make eggs, but it's associated, that same aromatase, with prostate cancer and breast cancer. Big concerns for humans. With regards to prostate cancer, the own company showed and published a paper showing that there's an 8.4-fold increase in prostate cancer in men who work in their factory bagging atrazine. And it's a community that's 80% black or 80% African American. With regards to breast cancer, there's a strong correlation in at least one study that women whose well water is contaminated with atrazine are more likely to get breast cancer, and that's just a correlation. But if you look at rats, atrazine causes testosterone to go down, just like it does in fish and frogs and reptiles and birds, and it causes a concomitant increase in estrogen. And those same rats, and the company's shown this, those same rats, when you give them atrazine, have a higher incidence of breast cancer that's estrogen dependent. The company published that, not me. In humans, we've shown, and another lab has shown as well, if you take a human cell and give it atrazine, this work was done with a graduate student, that cell will start expressing aromatase and making estrogen, just like we've shown in fish, just like we've shown in amphibians, just like we've shown in reptiles, just like we've shown in birds, just like we've shown in rats. This is now a human cell. I went to visit them. I still think they should spell their name with an I instead of a Y, but nobody listens to me. So they have a pipe that runs straight into the Mississippi River. 1.2 million pounds of atrazine flow into the Gulf of Mexico in an area that looks like this, and in an area that's 80% black or African American. I bring that up because, as part of the advocacy piece, if you look at the top 13 cancers that you're going to get in this country, shown in black, and red now, 11 of the 13 are ones that you're more likely to get if you're black, if you're African American. If you look at mortality relative to whites, to Caucasian Americans, you're more likely to die from 13 out of 13 cancers if you're black or African American. Now, my colleagues who study cancer say that less than 30% of cancer is genetic. That means that when your doctor tells you you're more likely to get breast cancer if your mother or your aunt or your sister or somebody else in your family has it, he or she's not telling you that it's genetics. They're telling you you've been exposed to the same crap that the rest of your family has. Because if you're a minority, you're more likely to live in and more likely to work in areas where you're exposed to chemicals that we know are associated with adverse health outcomes. I gave a talk for Coleman for the Cure. I called it an ounce of prevention. They don't give me no money, but they want to know what I had to talk about. And I'm all about that. But other than HeLa, I heard somebody talking about HeLa last night while I was eavesdropping over dinner. Other than HeLa cells, there are no cells from black or Hispanic women used in cancer research. So even if you find the cure, it might be irrelevant to the people who are more likely to get and more likely to die from those same cancers. The problem is you can't put a name on and you can't make money from prevention. I had a student who showed that if you give breast cancer cells atrazine, they start making aromatase and estrogen, just like we've shown in frogs, just like we've shown in fish, et cetera, et cetera. And that's significant, though, because usually you get breast cancer after menopause. So why are you getting an estrogen-dependent cancer when your estrogen levels in your blood are lower than they've ever been in your life? That's because aromatase expression locally in the breast is important. It induces estrogen, and that estrogen drives those cancerous cells and turns them into tumors. In fact, the aromatase expression is so important that a chemical called letrozole is the #1 treatment because it knocks out aromatase and decreases estrogen so those cells don't grow. Isn't that crazy? When the #1 contaminant of drinking water does the opposite, it turns on aromatase, increases estrogen, induces cancer in rats, and is associated with breast cancer in humans? Well, it turns out Novartis Oncology, before they changed their name to Syngenta, Novartis Oncology offers treatments for cancers that range from breast cancer. So the same company that gives you 80 million pounds of atrazine that induces aromatase every year also sells the #1 aromatase blocker for treating breast cancer. So that if you're a black or Hispanic or any woman living in the Midwest taking letrozole to treat your breast cancer, you're drinking a chemical from the same company that does exactly the opposite. I published a paper called The One-Stop Shop, Chemical Causes and Cures for Breast Cancer. The lawyers wrote me all kinds of crazy letters. But I'm just telling you the truth. At the year 2000, Novartis sold both chemicals, and then they split, coincidentally. So I think that my interest as a little boy who likes frogs and this aquatic organism has told me quite a lot about this aquatic organism. Because we develop in an amniotic fluid, and we know that your kids will be exposed to over 300 synthetic chemicals before they're even born, most of which we have no idea what they do, but we do know that we depend on testosterone, estrogen, thyroid hormone, cortisol, just like my frogs. And I would argue that one of my tadpoles trapped in a contaminated tank or contaminated pond is no different than a developing human fetus trapped in a contaminated amniotic fluid. Other studies, I'm one of the few scientists that brags about what I didn't do. Studies have shown that atrazine causes prostate and mammary cancers in rats, that it causes immune failure in rats, it causes neural damage when rats are exposed in utero. And as 3 studies published by the Environmental Protection Agency moved me in a very special way, much more than my own work. One is they showed that if you give a rat atrazine, it causes abortion. Another EPA laboratory showed that of those rats that don't abort, the sons are born with prostate disease. Another EPA laboratory showed if those rats that don't abort, the daughters are born with poor mammary development, such that when they grow up, their offspring have retarded growth and development. Imagine this. That rat on the bottom never saw atrazine. That rat on the bottom was affected by atrazine that its grandmother was exposed to. The rat on the bottom was affected by atrazine that its grandmother was exposed to. The rat on the bottom never saw atrazine. the rat on the bottom was affected by atrazine that its grandmother was exposed to. And that moves me in a very different way to think that my grandchildren could be affected by chemicals that we're using today. That makes me realize that I have a much bigger responsibility than just a little boy who likes frogs. It's already been shown that there's a correlation between birth defects and peak exposures to atrazine, and I don't have time to show you all of the papers and all of the effects, but the ones that are important to me, and because of my work, and I apologize, there's some graphic images coming, is this case-control study of maternal residential atrazine exposure and male genital malformations. The short story is this study, independent of me, showed that if you're pregnant with a male child, you're more likely to have a baby with hypospadias, where the urethra doesn't end all the way through the penis. You're more likely to have a son with cryptorchidism, where the testicles don't descend in the scrotum, and you're more likely to have a baby with microphallus, where the penis doesn't grow for life. And it interests me because male genital development depends on testosterone, and you can induce all of these birth defects with estrogen. And if you're given a chemical, those are facts I just told you, and if you're giving a chemical that reduces testosterone and increases estrogen in your fetus, it looks like it didn't have enough testosterone and had too much estrogen. We have to stop using the adult white male to figure out, and I show this guy because he doesn't like me, he works for the company. We have to stop using the adult white male to determine the safety of chemicals, because we know that something that might be safe for an adult white male can cross the placenta and affect the developing fetus. We know that even after that, these chemicals, including atrazine, can come out in the breast milk. And we know that even after that, a little bit of poison to an adult can be a lot of poison to a developing young child. I thought about all this stuff, and I thought about the advocacy issue. Syngenta has this on their website, that they say they have no obligation to update forward-looking statements to reflect actual results. Who the hell says shit like that, pardon my language? But what's more is in court documents that were just released, Syngenta had secret meetings. They had notes where they wrote here, for example, TBA, I'll tell you what that is in a minute. They wrote this in 2002, may be a bit more potent than atrazine. So they're admitting, even though they fought me in the public for the longest, they're admitting that they know it does something. They say lower doses cause same effects. They go on to say it causes mammary tumors. They go on to say it causes testicular. This is their handwriting. I told you I'm no chemist, but let me show you what TBA is. This is atrazine. TBA stands for terbutaline, and that's terbutaline. Differs by one methyl group. And you know what terbutaline is? In 2002, just a couple of months after they wrote that note in their meeting, terbutaline is what they released in Europe to replace atrazine. So somebody sat in a room, somebody presumably with children, certainly with parents and neighbors, somebody sat in a room and wrote, hey, this chemical's worse than atrazine. Let's give it to Europe. I thought about all that, but then I thought about what we get in academia. Don't be an advocate, my advisor told me. Let the science speak for itself. Don't be an advocate. Let the science speak for itself. That's what I was taught. That's what we're taught. And then I saw this from the EPA. They said about my work in 2006, me and my students' work, the ultimate decision, they said, about atrazine is much bigger than science. They said it weighs in public opinion. And then you know what I thought about? I thought about my mom. The EPA says they're weighing in, they're counting on my mom to have an opinion when she can't even get access to my work because I'm being promoted based on how many papers I publish in a magazine that 99.9% of the world has no access to. And I crossed the line. I crossed the line. I helped Keith Ellison write a bill to ban atrazine in Minnesota. That's why I got the day. I crossed the line when I helped the EPA understand my work and the others. I crossed the line when I participated in a number of documentaries, including one with Aaron Brockovich, including one with Jonathan Demme, and there we are in Salinas. And that's a student who grew up there and gave us the tour of where she and her family used to be sprayed. I helped a lawyer get $105 million out of those guys to get atrazine out of the water. And I participated in a number of popular articles like the New Yorker, that based on Facebook and based on views, had much, much bigger impact than my PNAS and Nature paper, which nobody else got to see other than you guys. I want to conclude with something that Syngenta said in the New Yorker article. They said, I am troubled by a suggestion that we have ever tried to discredit anyone. our focus has always been on communicating the science and setting the record straight, because they harassed me, and they followed me around, and they had secret meetings about me. And during that court case, their secret meetings, their notes came out. She says, I'm troubled by a suggestion that they ever tried to discredit anyone. Look what showed up in that case. Look under science. Look at the first thing they wrote down under science. And they called me crazy. The first thing is, you don't write something like that down. Jeez. I'd rather be crazy than stupid any day. It's caused me some trouble, though. Sometimes I go to speak, I get things like this. They tried to buy my name on the internet. They post these things on the web. And I thought long and hard about what I was taught as a graduate student. Don't be an advocate. Let the science speak for itself. I decided that I can't do that. You shouldn't do that. We have to change how we value public service and advocacy and teaching, because we all know what we really get paid for. I have a new philosophy now. Somebody once said, those who have the privilege to know. I didn't grow up privileged, but I've been to Harvard and Berkeley. I got a few letters. I'm certainly privileged now. Those who have the privilege to know, this person said, have the duty to act. So not only can you be an academic, a researcher, a scientist, a teacher, and be an advocate, but it's your duty, at least according to this guy. My time's up. Thank you. I know time's up, but I will take a few questions if you want. I'm sorry, I know the time's up. I'd never given a talk with that much sort of side stuff on advocacy and teaching, so it went up to the time. But if you don't mind, I will certainly take questions or comments, and if Syngenta's in the audience, whatever it is you might have. Usually they are. So the question is, what do I eat? I mostly, for atrazine, it's in the water. It's not going to be in the food, but there's 80,000 synthetic chemicals out there. What I ate up until the drought 2 years ago is my wife and I grew all of our own food. So, and I always feel uncomfortable saying that because we're in California, we have the space, we can do that, and not everybody can. But certainly eating organic and eating local is important whenever you can. Somebody told me to use the term voting with your dollars and where you spend your money, because organic doesn't mean chemical-free. We all know that, and it can mean different things in different states, but it's a message, and it's a message that I think we have to promote. Others? Somebody just yelled, because I can only see about that far. That's why I called on you. Yes? Oh, I hate this question. I'll tell you why. So the question is, can you filter atrazine out? Brita Filters even says on the side, removes atrazine, or at least it used to. And boy, if I had cut a deal 20 years ago, I'd probably be rich just from the Brita Filters that I sell. You can, but it doesn't solve the problem for the workers in the factory and the farms, because those high levels are mostly from inhalation and absorption across the skin. It also doesn't solve the problem for wildlife and the environment in general, and it also puts the burden on you when it really should be on them, because that's where that $105 million settlement came from, was water companies suing to make them remove it from the water for their customers. But you can. And just about any chemical pesticide would be filtered out. Yes?
How do you recruit your undergraduate students?
How do I recruit my undergraduate students? And the answer is I don't. So it's sort of a self-fulfilling thing. So I meet them through either classes or I guest lecture, or in many cases, they've talked about me in their other classes, or I'm in the Bio 1 textbook and the environmental toxicology textbook. So most of them kind of know what I do and things like that. In terms of creating a diversity of students, my biggest problem was recruiting adult white men. And I think part of it is once you have the reputation and once people know the lab and know the group, that people come. And so at this point, if anything else, I have to turn down students when I know that I'm having too many. Because my lab's not one where there's a graduate student who takes care of 3 undergraduates. I interact with all of my undergraduates direct. In fact, sometimes my graduate students say at their expense that I pay more attention to. But I expect my graduate students to be a little more dependent. So I work directly with the undergraduates and design the projects and put them in charge of the way I was treated when I was an undergraduate. So there's not much, at this stage, there's not much recruitment at all. My lab is known as kind of a big family place. I have a long over 20 years, over 200 students that one of my previous students now is planning something called the 20th Friendiversary. So we still hang out and do things. And so there's a long, extended sort of family from the lab as well, that students know people who go. And that's been the other thing that's really neat is when I have a student who wants to go into PA or physician's assistant, or one who wants to go into MD, or one who wants to go into law, or one who wants to go to graduate school, there's somebody in our extended family that they can talk to directly about their path and how they went from my lab to there. But I really don't do much recruitment at all now. So once you do the recruitment and once you get the reputation, it just sort of happens. What is that phrase? Build it and it will come, or something like that. I can't remember what that's from. Others? Okay, way in the back. I can't see the blue shirt. So the question is there a map of contamination? The U.S. Geological Survey does a wonderful job. So that map that I showed is a U.S. Geological Survey map based on sales, and that's done by county. So you can certainly, and I know I've done this in my home state, South Carolina, you can certainly look at usage based on sales, and there are maps where they've monitored pesticide levels, and you can look at those maps and then map that onto real estate property values and make those kinds of, there are a few papers that have already done that, but you're able to do it on your own if you have the time and the inclination. The data are out there. Yes? At least 2 of the metabolites are still active, and then you start to lose those effects. So it's a loss of, I don't have the chemistry up here to show you, but it's a loss of one of the side groups.
We have time for one more burning question, if there is one.
Either.
Just
somebody. I
pick one.
I don't know how to pick.
I just wanted, my name's Christine Broussard. I'm from the University of Laverne in Southern California, close to those areas of agriculture. And I just wanted to say thank you, because all of the good work that you've done in your own lab with your own students, you've also publicized the work that you do. You've put YouTube videos up, and as a cell biologist, developmental biologist, I'm able to use that in my classes and show students, you know, that we can be advocates as well as very good scientists, and that there's room in the world for a lot of diversity in the work that we do. So I just wanted to say thank you.
Thank you, but I want to give credit where credit's due. Other people have put up the YouTube. I do have a website that I haven't maintained for a couple of years, but other people do put those things up and advocate and things, and blogs and things like that as well. Yeah.
Please join me in thanking Dr. Hayes one more time. And we'd also like to thank all of you as you're departing the room. Just a quick note, please remember to complete your evaluations after our conference. We hope you've had a great conference experience. And we also like to remind you that completing the evaluation survey qualifies you for a chance to win either an iPad, for which we guarantee delivery by Christmas, or 2 complimentary registrations to next year's STEM conference, which will be held in San Francisco on November 3rd through the 5th. Again, thank you and safe travels home. We are adjourned.